Greetings, students. Mr. Labonte. I'm going to be talking about cells and primarily organelles in this presentation, but I'm also going to try to give you a sense of life and cellular life. So we start with our obligatory cartoon. I just wish you could see what the inside of your lungs look like. This is the smoking vacuole. All right. So I talked a little bit about this already in the class, but we're starting here by looking at the two basic cell types. There is prokaryotic and eukaryotic, and actually the U should be with, probably not after, but with the kernel and before the kernel. So the kernel in this case is the nucleus. So the two basic classifications are going to be without and with the nucleus. Some of the differences are in, obviously, the nucleus, whether or not it's there. But additionally, size. So this is about 1 micron, whereas this is 10 to 100 microns. So this is 10 to 100 times the size. Eukaryotes are 10 to 100 times the size of prokaryotes. There's a number of other components as well, but one that is super important is this idea that there are no membrane-bound organelles. So when we say membrane, we are talking about the PBL, that is the phospholipid bilayer, right? So prokaryotes have no phospholipid bilayers, and eukaryotes have a lot of membranes or bound organelles. So they have a lot of organelles that have the phospholipid bilayer. Cytoplasmic streaming is basically the movement of cytoplasm from one place to another, and I'll show you a little film of that in class where you can see a protist moving by cytoplasmic streaming. Chromosomes, there's a single chromosome, which is a loop, and histone we'll talk about later, but it's a protein that the chromosome are wrapped around. In eukaryotes, we have generally multiple chromosomes. So, for instance, humans have 23 chromosomes, right? And they're wrapped around things. And then the way they divide, this is through binary fission. Binary just means two, split into two. And the process for eukaryotic cells is mitosis, assuming it's not. All right, how did this come to be? Well, we started with a common prokaryotic ancestor. And it looked like very, very early on. So this is, we're looking really at like 4.2 billion years ago when we're looking at the start here. Very early on, we had a split from bacteria and archaea. So this is one split. Both of these groups are prokaryotes. And then from archaea, we had a split into eukarya or eukaryotic. The thing that's interesting is you are probably much more used to bacteria than you are to archaea. So for instance, something like E. coli, which populates you, all of the outside of your body, the inside of your body as well. That's bacteria. Archaea, these are the things that are found in thermal vents. So for instance, methanothermus, this is a prokaryote that eats methane and lives in thermal vents. So a lot of these, you see thermos, haliophiles. So these are really, these are often called extremophiles, so they love extreme environments. But it looks like that the eukaryotes actually evolved from them. So what's in eukaryotes? All animals, all plants, all fungi, and then all of these other single-celled organisms and kind of strange multi-celled organisms on this side, like slime mold. So basically everything that you recognize as life is eukaryotic. This is just another tree that shows you life and how life exists. That's bacteria, right? And then these would be animals, fungi, and plant. So there really is this kind of there's a lot of different ways to classify life so that you can see and figure out what they are. And I'm looking here if I can bring you down to what you're going to care about, of course, which is us. I can't see it off the top of my head in the animal groupings. All right. But now, if we're looking at cells, this is just basically, we're gonna jam this in our mind. If you haven't had this, lots of times people have had this information before, so this may be review for some of you. But if we're looking just simply at cells, then I'm going to help you define the different organelles that are in the cells, okay? For this, P stands for plant, A stands for animal. Okay, so that's what the P and the A stand for in this example. And then PR is going to stand for prokaryotes. So PR, you see PR here, that's a prokaryote, right? So that's before the nucleus. So by definition, none of these are going to have it. So this is going to be the name of the thing and the function of the thing, what it does. And so you can just see there's the nucleus, nucleolus, cytoplasm, mitochondria, Golgi apparatus, ribosomes, smooth ER. And that ER, by the way, that stands for endoplasmic reticulum. Plants and animals have that. Rough ER, lysosome, centriole, microtubules, microfilaments, vacuoles, chloroplast, plasma membrane, and cell wall, plants. And then, so I don't know what the E here stands for, so you can mark that out. Sorry about that. But one of the things I want to mention, and I'll highlight this later as well, but if I can get my eraser to work. I guess I can't. One of the things I wanted to mention, I'm going to use red here, is I'm going to just put a little red mark next to all of the things that have a nuclear that have a phospholipid bilayer. Okay, so nucleus, mitochondria, Golgi apparatus, smooth and rough ER, lysosomes, vacuole, chloroplast, and the plasma membrane. So when we talk about the phospholipid bilayer, that's a huge portion of what exists in the cell. So it is likely that you will have a quiz over this material. Your knowledge of it should be at the recognition level, so be able to like a matching kind of a thing. Be sure you have it to that level, but beyond that, it's okay. We'll work on it. Oh, excuse me. All right. So this is another way of thinking about organelles, and that's dividing them into groups. And the groups can be divided by pretty much, function, F-U-N-C-T-I-O-N, that's a T, by their function. So is the organelle's job to do protein formation? The nucleus, smooth and rough ER, Golgi bodies, and lysosomes. All of these have to do either with protein formation or protein manipulation. Lysosomes generally will destroy proteins. Does the job have to do with energy or food? Food storage or food waste. Mitochondria makes energy, chloroplast makes food, vacuoles store food. So you can organize it that way. Or does it have to do with structure or movement? Cytoskeleton, microtubules, microfilaments, and centrioles. All of these things have to do with structure and movement in a cell. So this is another way to kind of look at how a cell is organized and the mechanisms. Just another way to think about. Okie doke. We are going to go over this image in detail. We'll spend weeks on this particular image. But because it is probably the most important task in a cell, I wanted just to introduce it to you here. And this is the formation of proteins. So how are proteins made? The central tenet is you go from DNA to RNA, and then you go from RNA to amino acids, which then turn into proteins. So this is called the central tenet. And it's just the creation of proteins. So DNA is what you use to make the proteins, but you have to turn it into RNA, and then that RNA codes for amino acids. And this is the design of that. Why do we care so much about proteins? Because if I'm not talking about a phospholipid bilayer, I'm probably talking about a protein in the cell. They're super important. Pretty much all the things that happen are proteins. So for example, this is a ribosome. It's made primarily of proteins. The other thing that is super important in the cell is this basically phospholipid matrix. And I talked about this a little in class, but what I want you to see is, I'll just use this, let's see if I can come up with a nice interesting highlight color here. Try black. How pervasive phospholipid membrane is. So I'm starting right here. This is the nuclear envelope. So we're starting here. It turns in, this is now the rough ER, the rough endoplasmic reticulum. This is all one phospholipid bilayer. Now it breaks off and turns into a vacuole, but that vacuole will become part of the Golgi apparatus. That will break off, and then that Golgi apparatus will turn into a vesicle, which will then fuse with the cell membrane. So what you're seeing here is really almost a connection, not always physical, but a connection in synergy and similarity all the way from the nuclear envelope to the cell membrane. And all of these are phospholipid bilayers in their connection. So the other thing that I think is important for you to understand is that the shape of cells are a result of their job. So sometimes you'll see cells which are perfectly spherical or some, pretty darn close to spherical. These are globular cells, and usually they secrete things. So they have a large volume and a low surface area, and their job is to push things out. Otherwise, you might see a cell that looks like this, right? And this is actually a muscle cell. And the job of the muscle cell is actually to contract along a single axis. So that's the function of this particular cell. Other cells might have little arms that reach out like this. So these would be dendritic cells, okay? And a function of a cell like this is to figure things out or to reach around and to look out for certain things. So whereas when I draw a cell, I'll draw something like that, or I'll draw something like this to indicate that you have a particular cell. Really, these shapes almost never exist in an animal, and the cells in an animal are specialized due to their specific function. This means as well that you will have cells without organelles. So for instance, a red blood cell, or RBC, has no nucleus. a mature red blood cell has no nucleus. So even though we say, oh yeah, all eukaryotic cells have a nucleus, here's an example of a eukaryotic cell that doesn't have a nucleus. So cells are very, very specific depending upon the task that they have. Oh, great. And then actually I talk about these right here. So these are erythrocytes, or red blood cells without a nucleus. Here's a muscle cell, super long and thin, in order to constrict. And then these eosinophils, right, these are also white blood cells. They have tons of lysosomes. And lysosomes contain enzymes that dissolve. And the reason that they would do that is they would be taking in bacteria or other pathogens and destroying them. So another example of concentration of a particular organelle in a particular cell with a particular job. Okay, I hope that helps.